Okay, so we are going to review the action potentials that we talked about in the last section. So the generation of an active potential, number one, a local potential creates a threshold stimulus. Voltage-gated channels open and generate an action potential, causing a rapid sodium influx, depolarizes the neuron. The sodium channels close, and that decreases the movement of sodium. Then the voltage-gated channel, potassium channels open. That begins the repolarization of the cell. So remember, depolarization is where the cell membrane becomes less negative than its resting state. Repolarization, it starts to return to its resting state. The continued outflow or efflux of potassium causes hyperpolarization, and hyperpolarization is when the membrane potential is more negative than the resting state. So there's this great little video of action potentials that shows the graph. I love the graph. The graph really helps me think about it. So watch that 3-minute video on action potentials. It shows the opening and closing of the membrane channels, and it shows the graph of the millivolts, and you can kind of see what happens. So one of the things that promotes forward movement of the action potential, so it moves on down the axon, is what's called a refractory period. So you have two different types of refractory periods. The refractory period is where the membrane is not at its resting state, and so it's not electrically excitable at that point, or it's not as electrically excitable as normal because its channels are either open or the ion concentration hasn't returned back to its steady state. So there are two different types of refractory periods that we'll refer to. One of them is the absolute refractory period. The membrane is unresponsive to a new stimulus because the sodium channels, they can't be reopened for a specific amount of time following closure. So if we look back at the sequence of events, the sodium and potassium channels are closed, we get a threshold potential, the sodium channels open, sodium enters the cell and depolarizes the axon, and then the sodium channels close about a millisecond later, and the potassium channels open and we begin repolarization of the axon, and then we a lot of times get into that hyperpolarization where the cell is more negative than at its resting state. So during the time when the cell is way less negative than the resting state, that is the absolute refractory period. Those sodium channels are closed and they're not going to reopen. They sort of have to regenerate before they're going to open again. That's our absolute refractory period. Even if you got another stimulus right then, you absolutely would not fire the neuron during that absolute refractory period. During the relative refractory period, now the membrane potential is starting to return back to that minus 70 millivolts. It's returning back to its resting level. At that point, a stronger than normal stimulus could activate the sodium channels to open again. So you're starting to get into where you're even more negative than the resting state, but if you had a strong enough stimulus that took it way up here again, you could fire that nerve again. So during the absolute refractory period, you can't open the sodium channels. There's a specific amount of time before they're going to reset, basically. During the relative refractory period, if the stimulus is strong enough, you can reactivate those sodium channels. So there are several short little videos about refractory periods and action potentials. And the one on the bottom, the 5-minute one, reviews the whole process that I just went through, and it has the electrical graph and information about the ions. I really like that one a lot. So I would definitely recommend watching those videos. I like the animation because it kind of shows how it all happens. So the action potential has to propagate down the axon in order to spread along. So you get a little bit of the change in electrical potential spreading passively just to adjacent regions of the membrane. As you start to generate more and more depolarization, when it reaches a threshold, another action potential is generated and it continues to spread along the axon. So that's when we talk about there being a passive component and an active component. It spreads a little just because as those sodium ions are going in, it's becoming more positive. So the way it shows it here in this slide, it's like an enlarged section of the axon. The part where it's positive on the inside of the axon in letter A, that's where the sodium ions are rushing in. It can start to affect the area just right around it. It causes the interior of the axon to become a little more positive than when the membrane is resting, or a little less negative, which is how we've been saying it. When you get to the point where you activate those voltage-gated channels in the next section of axon, it'll regenerate an action potential and rapidly depolarize the membrane. So in that way it propagates down the axon, which is you know, how you keep it moving. In a myelinated axon, which is most of our axons are myelinated, the myelin acts as an insulator and increases the speed of the action potential and the distance that the current can passively spread. So each little Schwann cell has myelin wrapped around a section of the axon, and there are parts in between. If you look at the diagram here, there are parts in between that are called nodes of Ranvier, and there are more voltage-gated channels in those sections that are not myelinated, and the charge actually hops from node to node, and that speeds it up as well. It's called saltatory conduction, that hopping from node to node. The action potential appears to jump from node to node along the axon. So when it crosses that unmyelinated node, there are more voltage-gated receptors there, and it moves the charge along. So there's a little video on the saltatory conduction, which is kind of neat. It's a little animation. Obviously you can't take real videos of these because you can't see electrical charge, but that saltatory conduction, that's why myelinated axons transmit nerve conduction faster than unmyelinated. Okay, so some axons are faster at progressing that action potential because either they're larger or because they're myelinated. So they use the example in the book of a water and a hose. If you have a big wide hose, like a fire hose, it's going to pour more water in less time than like a little garden hose. And so a larger diameter axon is going to propagate action potentials faster. They compare the myelination to tape around a leaky hose, concentrating the flow of water. So if you have those myelin sections, it lets the electrical charge propagate without leaking out, if that makes any sense. So the myelin, of course, has a lot of fat. That's why it appears white. It's in the white matter. It is that sheath of protein and fats that's created by the Schwann cells, and we'll talk about Schwann cells in the next section. It provides that insulation and it prevents current flow across the membrane. So it increases the speed of the action potential propagation, and it increases the distance that the current can passively spread. Thicker myelin leads to faster conduction and greater chances for action potential propagation. So the nodes of Ranvier are the patches on the myelinated axons that lack myelin. It's basically the space in between two Schwann cells. And they're about every 1 to 2 millimeters along the axon, And remember, we said that passive propagation went 1 to 2 millimeters. How convenient. So it can passively propagate and hop those little nodes. They contain really high densities of sodium and potassium channels, and so that electrical charge right away opens those gates and it propagates the action potential down the cell. So the action potential spreads rapidly along a myelinated region, and it slows down a little bit when it's crossing that node of Ranvier. And the saltatory conduction is the quick node-to-node jumping of the action potential down a myelinated axon. Okay, so we talked about the functional groups before: the afferent neurons carrying sensor information from the outer body towards the central nervous system, the efferent neuron relaying commands from the CNS out to the muscles and glands, and the interneurons throughout the nervous system processing information locally or conveying information short distances. So of all of those, we have the most interneurons. So when we talk about, when we were talking about the different types of neurons, where we had the multipolar and the bipolar and those sort of things, there's a lot of reason for that, and it's because of the different types have different interactions between the neurons. So we talk about divergence and convergence for distributing information throughout the nervous system. So convergence is when you get multiple inputs from a variety of cells on a single neuron. So you picture those multipolar neurons that have all sorts of different dendrites, and they're getting tons of input from different neurons. Divergence is where a single neuron has a lot of branches that terminate on a lot of different cells. So there's a little diagram here, and of course this is figure 212 in the book. It shows convergence, where you've got in the picture here, there are 3 different neurons synapsing on one little neuron, so all 3 of those are giving information to that one. In the second picture, it's divergent, where you have one neuron with 3 little branches diverging out to those 3 separate neurons. So those could all be 3 separate pathways. They're going to do 3 separate things. And that's just a little bit about how those different types of neurons interact with each other. Okay, so I'm going to wrap this up, and next time we're going to talk about the glia, or the supporting cells of the nervous system.